Modern genomics confirms every European carries Neanderthal DNA, between 1 and 4%. But that number is not distributed equally. Specific archaic variants tied to blood coagulation, immune defense, and physical robustness persist at sharply different frequencies across lineages. Historians explain the divide between rulers and the ruled through politics, wealth, and warfare. Geneticists may have found something older in royal bloodlines, something no one is rushing to explain. To understand what it is, you start where the evidence starts, inside the human genome, and then inside a grave sealed for 2,500 years. Genome-wide maps of Neanderthal ancestry reveal a landscape that is anything but unequal. While every person of European descent carries archaic DNA, the distribution of these segments is sharply uneven. High-resolution frequency plots from the 1,000 Genomes project and IBD mix analyses show regional spikes. Certain Neanderthal-derived alleles reach 20 to 30% in some populations, while barely registering in others. These are not random relics. Some of the most persistent variants influence traits tied to survival, blood coagulation, innate immunity, skin pigmentation, and even adult height. The F7 locus, for example, carries a Neanderthal origin allele that increases plasma factor 7 activity, found in up to 12% of West Europeans, but almost absent in East Asia. The OAS cluster, linked to antiviral defense, sits at 20% in Northern Europe. Meanwhile, pigmentation alleles like those in BNC2 and SLC45A2 trace a north-south gradient, echoing the ancient push and pull between ultraviolet adaptation and vitamin D synthesis. These patterns are not explained by geography alone. Genome-wide selection scans detect signals of positive selection. Long stretches of DNA where archaic variants rose in frequency faster than chance would allow. The story these maps tell is not one of passive inheritance, but of an ongoing contest where some genes survived because they offered an edge. Yet the meaning of these uneven frequencies is far from settled. Interpreting what they signify, biological difference, historic accident, or something more, requires careful definition. Before any claim about bloodlines or power, the data demand a closer look at what exactly is being measured. Introgression and positive selection are the twin engines that drive much of the story hidden in our DNA. Introgression, in the language of genetics, is the movement of genes from one population into another, a process that for humans began when early modern humans met and interbred with Neanderthals more than 40,000 years ago. Their genes did not simply vanish into the background. Instead, fragments of Neanderthal DNA were taken up, copied, and passed down, becoming a permanent part of the genetic fabric in people outside Africa. But not all archaic DNA persists equally. This is where positive selection enters the picture. Positive selection is the process by which certain genetic variants, alleles, rise in frequency because they offer a real advantage. If a gene helps its carrier survive disease, withstand cold, or process new foods, it becomes more common with each generation. Over time, these alleles can sweep through a population, leaving behind long stretches of DNA that stand out against the randomness of genetic drift. The result is a genome that is not a smooth blend, but a patchwork. Some regions shaped by ancient encounters, others by the relentless logic of survival. Introgressed Neanderthal segments are found in genes that affect blood clotting, immune response, and skin color. When selection favors one of these archaic variants, its frequency can jump from a few percent to a substantial minority, or even approach half the population in some regions. This is not passive inheritance. It is a running contest. where only the most useful fragments are preserved by the pressures of history and environment. In this framework, the uneven presence of archaic DNA is not just a record of who met whom, but a living trace of which traits mattered enough to be kept. The terms introgression and positive selection are more than scientific jargon. They are the vocabulary needed to understand how biology, chance, and circumstance have shaped both ordinary people and those who claimed power over them. Genes operate outside the rules of kingship, yet their influence can tip the scales of survival. A single variant, arriving in the right person at the right moment, could mean the difference between bleeding out on a battlefield or living to see another day. Neanderthal-derived alleles, still present in modern Europeans, shape blood coagulation, immune defenses, and skin pigmentation. The F7 gene, for instance, affects how efficiently blood clots, a trait with obvious stakes in an era before surgery or antibiotics. OAS cluster variants sharpen the immune response against viral infections, while BNC2 and SLC45A2 shift how skin copes with sunlight, altering the body's adaptation to northern climates. These aren't just molecular quirks. They touch the heart of survival, health, and sometimes power. But did these biological advantages align with the social order? Genome-wide association studies show that archaic alleles tied to resilience and health are scattered unevenly, sometimes clustering in families or regions that wielded influence. The machinery of social hierarchy, dynastic marriages, guarded bloodlines may have acted as a filter, concentrating certain traits among elites, while the broader population followed a different genetic course. Medieval rulers didn't choose heirs by genetic code, but selection pressure worked quietly, favoring lineages that survived famine, war, or disease. Over generations, biology and circumstance could reinforce the myth, and sometimes the reality, of difference in royal blood. The next step moves from DNA to bone. Do the remains from Europe's grandest burials hold evidence that power left a biological mark? The Hochdorf chieftain's bones tell a story that complicates any easy separation between legend and measurable fact. Excavated from a burial mound in Baden-Württemberg and dated to around 530 BC, this individual stood 1.8 meters tall, nearly 6 feet 2 inches. For Iron Age Central Europe, where the average male height hovered at 1.68 meters, this is not a minor difference. It is a biological outlier that demands explanation. The skeleton's long bones reveal not only length but remarkable robustness, with cortical thickness suggesting years of high protein intake and physical vigor. There is no evidence of chronic infection or trauma. The dental record shows just 2% caries and minimal enamel hyperplasia, both rare for the period. Isotope analysis of bone collagen draws the profile into sharper focus. The nitrogen signature, delta 15N, at plus 11.3 per mil, sits well above the local mean, pointing to a diet dominated by terrestrial animal protein, meat and dairy, not the grain-heavy fare of commoners. The carbon isotope value, delta 13C, at minus 21.2 per mil, confirms this was not a diet built on millet or imported C4 plants, but on locally sourced animal products. Strontium ratios match the geology of the region, ruling out the possibility that this was a foreigner imported for display. Genetic sequencing, though incomplete, adds one more layer. The chieftain carried the IGF1R RS228839 allele, a variant associated with increased stature, at a frequency that matches the tallest ancient populations of Western Iberia. His Y chromosome falls within haplogroup RL51, a lineage found repeatedly among Hallstatt-era elites. Yet the presence of these alleles in other populations tells a subtler story. Genetic factors alone did not make the Hochdorf chieftain a giant. The combination of inherited potential, privileged diet, and low disease burden set him apart from the ordinary men of his time. What emerges from the burial is not a statistical curiosity, but a concrete signal of how power and biology could intersect. The grave goods, golden talk, bronze couch, imported drinking vessels speak to status, but the skeleton itself is a record of selective advantage, preserved not by intention but by circumstance. The Hochdorf chieftain's extraordinary stature is not explained away by random variation. It is a data point that demands to be read alongside the social structures that produced it. The next figure in this pattern, a woman buried with equal splendor at Vix, will test whether this outlier status was truly confined to elite men, or if the pattern runs deeper, across gender and geography. In the hills of Burgundy, a grave unlike any other was uncovered in 1953. The burial at Vix contained not a chieftain, but a woman, laid to rest around 530 BC, surrounded by treasures that rivaled any princely tomb of the Iron Age. The centerpiece, a bronze crater nearly as tall as a person, stood beside weapons, jewelry, and imported goods from across the Mediterranean. Yet it was the skeleton itself that demanded attention. Osteometric analysis placed her height at 1.71 meters, over 5'7". For an Iron Age woman, this was extraordinary. The regional mean for female stature hovered at 1.57 meters. The Vix woman exceeded it by more than 2 standard deviations, a Z-score of plus 2.33. In modern terms, she stood as far above her peers as a professional basketball player towers over a crowd. Her bones told a story of privilege, but also of biology. The femur's cross-section showed robust cortical thickness, the kind seen in individuals with a lifetime of ample nutrition and physical activity. Stable isotope readings, delta 15N at plus 12.5 per mil and delta 13C at minus 19.8 per mil, pointed to a diet rich in both terrestrial animal protein and marine fish, foods that rarely reach the tables of ordinary people. Dental wear revealed moderate attrition, but no signs of chronic malnutrition or infectious disease. Even the healed rib fracture and minor osteoarthritis spoke to a life of movement, not deprivation. Genetic sequencing recovered from her remains added another layer. The Vix woman carried the HMGA 2RS4828032 variant, a genetic marker associated with increased stature. Her mitochondrial haplogroup, H1E1A, traced back to Neolithic Central Europe, hinting at deep continuity between the region's ancient farmers and its Iron Age elites. Comparative data from other elite burials, Magdalenenburg, Ljubljana Hill, the Brunel Barrow, showed a repeated pattern. Individuals with both genetic predisposition and privileged diet, consistently measuring well above their contemporaries in height. In each case, Z-scores for elite burials exceeded plus 1.4, a statistical signal that cannot be dismissed as random chance. The Vix burial disrupts any notion that biological exceptionalism in early European elites was reserved for men. Here, the same triad appears: tall stature, robust health, and a diet out of reach for most. The pattern crosses both sex and geography. It raises the question: were these traits simply the byproduct of status, or did the machinery of power, marriage, and inheritance act as a filter, concentrating certain biological features in a closed circle over generations? The bones offer no intent, only evidence. But the convergence is impossible to ignore. As the inquiry moves from bone to blood, The search for difference continues, not in artifacts, but in the very chemistry of life. The phrase blue blood first surfaced in the courts of medieval Castile, where noble families traced their lineage back through centuries of carefully documented marriages. In those chronicles, sangre azul was more than a boast. It referred, quite literally, to the blue veins visible beneath the pale skin of the aristocracy, a physical trait that set them apart in a land of deep sun and darker complexions. Physicians of the early modern period, like Alonso de Ercilla, noted the phenomenon as an optical effect. Low melanin in the skin allowed short wavelength blue light to scatter from subdermal veins, making them stand out against the pallor of elite faces. This visible difference did not arise from myth alone. Genome-wide association studies now point to a cluster of pigmentation alleles, most notably the Neanderthal-derived RS1964425C variant in the BNC2 gene, linked to lighter skin in Europeans. The frequency of this allele can reach 80% in northern populations, falling sharply toward the south. Its presence is not random. Selection scans reveal a strong signal in regions where sunlight is scarce, suggesting that lighter skin conferred an advantage for vitamin D synthesis. Yet the story grows more tangled as it moves from phenotype to social meaning. The blue veins of aristocrats became a symbol of purity, separating those with noble blood from those whose ancestry was less visible. In this way, a measurable biological trait, rooted in ancient introgression and shaped by selection pressure, became a marker of social hierarchy. The metaphor of blue blood carried a narrow but real observation: that centuries of endogamy and selective marriage could, under the right conditions, concentrate visible and perhaps beneficial traits within a closed circle. But the same mechanisms that preserved these features would soon reveal their cost, as the royal obsession with bloodline purity collided with the harsh mathematics of heredity. A single error in the genetic code, one base out of billions, entered the royal bloodstream in the 19th century. Queen Victoria, through a spontaneous mutation in her F9 gene, became the unwitting founder of Hemophilia B in the European dynasties. The mutation C.316G to A changed a single amino acid in factor IX, crippling the body's ability to clot blood. Victoria's son, Prince Leopold, was the first to bleed uncontrollably from a minor injury, dying at age 30 after a fall. The same mutation soon threaded its way through the royal houses of Spain, Germany, and Russia, carried by daughters and granddaughters whose marriages were arranged for politics, not health. Irony is stark. The same closed marriage circles that preserved the illusion of noble purity also locked in a lethal defect. By the early 20th century, the royal pedigree chart reads like a medical case report. Prince Alexei Nikolaevich, heir to the Russian throne, bled internally from the same mutation that had killed Leopold. The Spanish and German branches faced their own losses. Young men, heirs, and symbols of dynastic continuity, brought down not by war or intrigue, but by a silent error in their DNA. Geneticists, led by Evgeny Rogaev in 2009, traced the mutation directly from preserved royal remains. The molecular fingerprint is identical across branches, confirming a single origin and a cascade driven by endogamy. The cost of status, measured in suffering and lost futures, became inescapable. The story of royal hemophilia is not a footnote, it is a warning. Isolation can concentrate both strength and weakness, and the very walls built to guard privilege can trap disaster within. As the documentary turns to the Church's marriage laws, the question sharpens. Did the machinery of power create a biological divide that shaped not just history, but the very blood of those who ruled. By the 8th century, the Catholic Church had stretched the boundaries of forbidden marriage to the 7th degree of kinship, a net so wide that even distant cousins fell within its grasp. The official canon, codified in documents like the Collectio Canonum Gratianis and reinforced at councils such as Tribur, made these prohibitions explicit. Yet the rules were not absolute. For the right price and the right family name, a loophole appeared in the form of papal dispensations. Chancery records from the papacies of Innocent III and Boniface VIII detail a brisk trade in exemptions, with noble houses paying sizable fees, sometimes 20 gold florins or more, for permission to marry within the prohibited circle. These dispensations flowed almost exclusively to royalty and high aristocracy, while ordinary people were left to obey the outbreeding mandate. The result was a system where the masses grew more genetically mixed, but the elite preserved their own lineage, generation after generation. Population genetics models suggest this dual track could drive a measurable wedge, raising inbreeding coefficients among the privileged, amplifying both their advantages and their risks. The Church's universal law, bent by privilege and money, became a mechanism for biological separation, one whose long-term consequences would echo through the dynasties of Europe. Charles II of Spain was born into a dynasty that had spent centuries circling its own bloodlines. By the time he reached adulthood in the late 17th century, the Habsburg pedigree had become a closed loop, traced and retraced across generations of cousin marriages and papal dispensations. Genealogists calculate his inbreeding coefficient, F, to be 0.254, a number that exceeds the theoretical offspring of brother-sister unions. The consequences were written in his body: pronounced mandibular prognathism, a weak immune system, infertility, and a lifetime of neurological and physical ailments. Contemporary accounts describe a king who struggled to speak, to walk, to produce an heir. Medical records and posthumous analyses point to a cascade of recessive disorders, each the product of identical gene copies inherited from both parents, an outcome that population geneticists recognize as inbreeding depression. The Habsburg experiment, driven by the logic of dynastic survival, ultimately collapsed under its own genetic weight. Charles II died childless in 1700, ending the Spanish line. His life stands as a terminal case, not of deliberate design, but of the relentless mathematics of heredity. The story does not resolve into a simple lesson. It leaves behind a question. When social systems close in on themselves, what else do they preserve, and at what cost? Today, positive selection still shapes our bodies, quietly, relentlessly, whether we understand it or not. The royal bloodlines, with their Neanderthal echoes, towering skeletons, and inherited vulnerabilities, map a centuries-long experiment in human biology. No medieval ruler knew the word DNA, yet selection pressure needs no intention. The real question: what divides persist inside us, carried forward by history's blind hand? Add your voice. What do you see in the evidence?